Okay, we're continuing with a chapter on ecosystems. And the previous slide, we talked about primary production and energy flux through a system. And this is a curve an Allen curve named after the fellow who figured it out, about how you can figure out what happens to the amount of production that's in the system of animals. And what happens generally is that animals have a lot of offspring, right? And then a proportion of them die every X number of days or months or whatever, right? So there's this variable survival rate. And particularly many organisms, humans don't have that high of a proportion relative to like a large fish that put out hundreds of eggs, right? Thousands of eggs, and then hundreds of them hatch, and eventually, maybe 10 of them survive. And so what we have on here is the mass of individuals, right? And we have the mortality in here, and the number of individuals. And n sub t would be the number at time t, and then n sub t plus 1 would be the number at time t plus 1, whatever time that is, t plus 1. So if there's a decrease over time from time period 1 to time period 2, right, that's mortality. Decrease in numbers is going to be due to mortality. That could be disease, predation, whatever. And then the biomass of the survivors, because anybody that makes it, probably, although it could be a stressful situation or not, but for the most part, the ones that did survive would be able to grow over that period of time. And so this here could be the biomass of those that died in the time interval gets taken out, right? And so what we see is the number of individuals versus individual mass is decreasing here. This brings up what was related to this, and that is called the Allen paradox. And what he did is go out to the stream and measure the total biomass of invertebrates, and then he looked at the number of invertebrate fishes invertivore fishes in there. And he said, look, if these fish eat at this rate, they're going to use up all the invertebrates that are in the stream in a few months. So how can they live for years in the stream? So the question of the day for now is, what is it that explains the ability of those fish to continue getting food even though they would eat up the total number there within a few months if you just use the rate they're consuming things? So the answer that he came up with was that biomass is not the same as production, right? So there's the invertebrates can reproduce and grow. Some of them are lost to mortality, but they continue to replenish that biomass as they reproduce and grow, right? So here's the mass of individuals. The biomass of the survivors is actually increasing, right? And it's going to make up for decreased numbers that are eaten. So again, we've had this same point that I made in the previous lecture, and I've made also when we talked about nutrient dynamics. If you look at the standing stock, it's an amount. That is not the same as a rate. So I set you up in that question. I said, look, they're eating this amount per unit time, but there's only this amount. The problem is that amount is growing per unit time. Stated differently, if the fish didn't eat the invertebrates, they'd either run out of food themselves or they'd grow and fill the stream, right? Because that's what's holding the levels at where they are. biomass is not a good way to determine secondary production, is what Sean says. That's correct. Right? Rates, growth, rates, and reproduction are not the same as the amount that's there. You can have something that's small, an invertebrate that's small and reproducing like crazy, like a chironomid midge. And it is, a lot of it gets eaten, but it keeps cranking along. You can have something that's big and slow, like larger predators, a very large bass, right? Or a catfish that's 30 years old and weighs 60 pounds. It's not going to, necessarily be growing very fast, but it grows slowly. That's also why it's easier to deplete large fishes from a fishery, because they tend to grow more slowly and not reproduce as quickly. Right. All right. So remember, biomass is not the same as amount. Rates are what we're talking about here. When we're talking about ecosystems, we're talking about two things. We're talking about a compartment and a rate. So here's an example of a nutrient flux budget. And this one is given from my doctoral research on a pond in Central Oregon, just on the east side of the Cascades, south of Crater Lake. It's called Mare's Egg Spring, and I went and measured all of the fluxes. This one has this flux budget is talking about the fluxes. So we've got compartments, particulate and dissolved in, the nostoc, the cyanobacterium that's a producer, the snails, sediment, caddis larvae, logs. And then we have the spring itself, which you can view as its own compartment, right? So inside the pool and outside the pool. So we can get at all the fluxes that come in here and get an idea of how the system works. So here's a question for you. Look, we've got sediment here, and then we've got this loss, this flux of loss to atmospheric end. So how do you think nitrogen's leaving the sediment and turning into N2 gas? What process is that? And in contrast, we have atmospheric N2 going into the cyanobacterium, and there's another one going into logs. But so this is an opposite. There's another nitrogen flux that I'm thinking about that's happening here. So anybody offhand have a guess of what those fluxes are? Nitrification is the conversion of ammonium to nitrate, right? So Aaron's right, denitrification. And Abigail's right, the cyanobacteria are fixing nitrogen. Also, there are nitrogen-fixing organisms living in the anoxic area of the logs. And so they're pulling nitrogen gas in, atmospheric nitrogen gas in. Then there's a series of other things that are happening here. So snails are putting nitrogen into the sediment. How do you think they're doing that? So are caddis larvae putting things in nitrogen. So yeah, Emma, pooping, or what's the other option? Slobbering. maybe sloppy feeding. That's possible. And how else do you turn into detritus? And this would be nitrogen containing, dying, right? Pooping, slobbering, and dying, exactly. So caddis larvae and snails are pooping, slobbering, and dying. The snails are, we already talked about this previously. The snails are actually scraping periphyton off of the nostoc, and they're getting that nitrogen from the nostoc. I wasn't able to get that rate in, but presumably it roughly that matches the rate out. So how do I get this? I put snails in a jar and I measure how much poop there are, how long, and I put snails in a jar and I measure how much nitrogen gets dissolved in the water, how much nitrogen they're putting out in terms of excreting ammonium and nitrate and such. And then there's the whole system stuff in addition to the atmospheric end coming in and out. There's losses, sediments just get buried from all aquatic systems. There's nutrients nitrogen that dissolved in the water that's flowing in. This is a spring-fed system, so it's falling up out of the bottom through the sand. And then it's going over a beaver dam and flowing out. Okay, and these are pretty closely matched, but they're not exactly. So all in the end, everything has to match, right? Your sheet has to balance. So that's how you know that you're doing things right. Some of these were a little bit difficult to estimate. So log infall, you know, I could see how big the logs were in the water. I could measure how much nitrogen there was in a new log that fell in. But I worked there for 3 years, so I had to figure out, you know, how often do logs fall? What's the probability they'll come in there, right? So, you know, pretty broad guess. Litter infall is pretty straightforward. You just put up some kind of mesh trap and catch everything that falls in for a year or so and measure how much nitrogen's in it. Or you can put up a pan and measure how much precipitation, nitrogen comes in the form of precipitation. What's this emergence here? Can anybody tell me what that is? And I think, who did the Missouri paper? That's the person that should tell me. No. So caddis larvae, what happens? The two aquatic invertebrate, they're larvae. Who was it that did insects fly away? Caddis leave their casing behind. Yeah, they're emerging as adults, right? And going out and reproducing and then laying eggs and coming back in. So there's this loss. Remember the Missouri paper, they talked about how much the birds depended upon the stuff that was coming out of that spring. There's a nitrogen flux. And then beavers are ecosystem engineers. They tend to go out into the terrestrial and cut down trees and either bring the trees in or eat the bark off the trees. So they'll bring a bunch of trees in and small saplings and stick them down underneath the water, and then we'll go back and strip the bark off and eat the cambium. And they then are moving nitrogen from outside the system into the system. In addition, they're putting a dam on the end, and that's keeping, that's also kind of changing the flux rates. And then putting that dam, they're pulling a bunch of wood into the system as well. This was a tough one because I had no way to know how much beavers poop, right? Or slobber, as Emma said. But there is some old literature on how much beavers eat and how much they excrete from zoos, because they have to figure out how much food to feed a beaver to keep it alive and going, and how much nitrogen they need and all those kinds of things. And so I use those numbers to estimate that how much they would be excreting. Supposedly beavers come in pairs for mostly, but I only saw one. So this is actually 1.5 beavers excretion worth. I wasn't quite sure how to set that. But they basically poop and put their basically almost wood pellets, and it just goes down. So they tend to eat outside, come in, and excrete in the pool. So this is an example of using nitrogen and fluxes into and out of pools to get an idea of what's controlling the nitrogen budget in the system. The thing that I thought was that the system was covered with nostoc, that's a nitrogen fixing species, and I thought, well, it must be really important in the nitrogen budget. Well, it turns out, yeah, sort of important, right? Because if you look at the total outflows, we're looking at about 5,000 here. They're only 300, so not even 10% of the nitrogen coming in the system is from nitrogen fixation. It could be more important, though, to some of the organisms, because the snails are eating the stuff that lives on the nostoc. So if the nostoc leaks out nitrogen, the diatoms and such take that up, then they're dependent upon nitrogen that comes directly from nostoc, and not so much on the stuff that's flowing through the system. Okay. You remember also that I showed a picture in an old lecture of a fluorescence micrograph where I had really bright red on top of brick red micrograph. That was the diatoms the snails eat, sitting on top of the cyanobacterium, which then has a different fluorescent signature related to its pigments that the phycobionts are allowed to use green lights. Okay, so we've already talked once about biodiversity and ecosystem function, and I just wanted to bring it up again. And one of the things that I didn't talk about very much when we talked about the biodiversity ecosystem function is when they go into ecosystems, even the groundwater ecosystems, for the most part, you can almost always use DNA analysis or RNA analysis to find out that every major group of organisms is there. The nitrifiers, the denitrifiers, the sulfur oxidizers, the methanogens, the methylotrophs, all of those fluxes that we talk about. You can't really find many systems that don't have those. The only potential exceptions to that are the deep groundwater systems that are extraordinarily simple and just have a couple of microbes, species associated with them that are apparently cycling all their nutrients. And that could well be a system where biodiversity is limited. That said, if you take some organic carbon like leaves and throw them sterilize them and throw them in a test tube and throw a bunch of different species of bacteria in versus one or two species of bacteria, the leaves will degrade faster when the whole bunch of species are there because they're able to specialize on all those different compounds. But eventually, the carbon will all break down, even with just a few species for the most part. In addition, benthic invertebrate diversity may alter nutrient flux across the sediment water interface, and also across hypolimnion in a stratified lake. This is because diffusion is limited to and from the sediments. Why is diffusion limited to and from sediments? And think about small spaces between particles and one of the properties of water that we talked about early on, and the type of diffusion that might happen. Boundary layer, excellent, right. So small spaces, low Reynolds numbers, high viscosity, low turbulence, low transport of materials through the boundary layer by eddy diffusion. So driven by molecular diffusion, unless there's invertebrates. I think I might have a picture. No, I don't have. There's a picture in the book about that.